pathogens are all around us. They are the viruses, bacteria, fungi, and protozoans that invade our bodies and make us sick. Your body has many defenses against pathogen invaders. Our skin forms a barrier that few microorganisms can penetrate. Your respiratory system is lined with ciliated mucous membranes to sweep bacteria into the back of your throat where they can be swallowed. Exocrine glands near the eyes and mouth secrete enzymes to destroy invaders before they have a chance to enter the body. Lymphocytes circulate through your bloodstream, organs, and lymphatic system. Although each lymphocyte has a specific task, all arise from the same stem cells in the bone marrow. Lymphocytes enter damaged or infected tissues by squeezing between the capillary endothelial cells. The primary function of a macrophage is to engulf and destroy foreign invaders. Macrophages can also alert other lymphocytes to the pathogenic invasion. B cells produce antibodies that bind to bacteria invaders and signal for their removal. To stop the spread of infection, cytotoxic T cells destroy body cells infected with viruses. Helper T cells stimulate the production of B cells and cytotoxic T cells. Suppressor T cells slow or prevent the immune response from happening. Memory B and T cells are produced during an initial encounter with a foreign invader. Memory cells circulate through the bloodstream to respond when the foreign invader attacks again. A vaccine immunization deliberately introduces a killed or weakened pathogen into your body to provoke an immune response and produce memory cells. A second injection, called a booster shot, elicits a secondary immune response. This produces more memory cells and antibodies to make you immune to the disease. Just as you may write your name on your belongings to identify them as yours, your body recognizes your own cells by MHC markers. These markers lie on the surface of your cells. Nothing else has markers exactly like yours. When bacteria enter your body through a cut, they are immediately recognized as invaders, which begins the antibody-mediated response. The invasion first triggers an inflammatory response. Next, macrophages move to the site of the infection and engulf a few bacteria cells. Inside, the macrophage's enzymes break apart the bacteria. Antigen fragments of the bacteria are transported to the surface of the macrophage, where they are combined with an MHC marker and displayed. Antigens are any large molecules with a distinct configuration that can trigger an immune response. Some bacteria slip past virgin B cells that haven't any membrane-bound antibodies. Other bacteria may encounter a memory B cell that has a membrane-bound antibody. Antibodies are Y-shaped protein molecules with binding sites for specific antigens. These B cells attack the bacteria and express the antigen as an antigen MHC complex. Stimulated by the macrophage's expression of antigen MHC complexes, helper T cells interact with sensitized B cells, causing them to divide rapidly. Some B cell progeny secrete antibodies, while others become memory B cells. Freely circulating antibodies bind with the antigen-covered invaders, marking them for destruction. More histamines are released, causing the inflammatory response to increase. This ultimately brings in more macrophages to engulf the invading bacteria. When a virus or other pathogen penetrates your cells, it may be hidden from circulating antibodies. This type of invasion triggers a cell-mediated immune response. During a cell-mediated response, cytotoxic T cells destroy your infected cells before the viruses can replicate and spread. When a virus infects a cell, viral protein antigens become associated with MHC markers on the surface of the host's plasma membrane. Cytotoxic T cells recognize the antigen MHC complex, bind to it, and secrete protein perforins that break apart the host's plasma membrane. Without drug treatments, tissue and organ transplants are rejected because cytotoxic T cells do not recognize the MHC markers of the transplant tissue. The transplant is perceived as foreign and triggers a cell-mediated immune response.